All right, welcome back to my kitchen this time. So last time, I don't know if you noticed, there's a cat in the background. So anyway, we're in my kitchen now, and we're going to talk about cell transport mechanism. So we talked a little bit about cell membrane, right? Remember the cell membrane? Had these phospholipids, right? And we had the hydrophilic and the hydrophobic regions. So when we talk about cell transport, we're just talking about how things, get across the membrane. So anyway, here we go. So important things that we have here. So diffusion, right? So diffusion is passive transport. So if you remember, diffusion goes from a high concentration to a low concentration, right? So if we have a cell membrane, right, and we have high concentration, and inside the cell we have low concentration. According to diffusion, the substance will diffuse in until equilibrium. Right? So when the concentration is the same on both sides, right, then you'll get some sort of equilibrium there. All right. So when we talk about diffusion, right, what kind of things diffuse? Right? So gases. Oops. Let me get out of here again. So gases. Let me draw that again for you. And small. And small nonpolar molecules. So those are the kind of things that diffuse. Polar molecules will not diffuse. All right. Polar molecules won't diffuse because they can't get through the middle of that phospholipid membrane. Right. So it'll diffuse from a high concentration to a low concentration. So we had facilitated transport. So we talked a little bit about facilitated transport before. Right. So facilitated transport is diffusion with a carrier molecule. So if we look down here, right, so facilitated transport, some sort of carrier molecule opens up a membrane and then it just allows things to diffuse through. So this, for most things, is kind of generally the rule because that makes it more selectively permeable. Gases like oxygen, we need oxygen to diffuse, so straight diffusion for that. But for ions, facilitated transport's kind of the rule for that. All right, so osmosis. Remember, osmosis is diffusion of water, right? So a couple things with osmosis, right? So osmosis, if we think about it, right? So this is our diffusion. Right, of our water molecule. All right, so that's diffusion of water. So it's aided by aquaporins, remember? And those are channels, right? So it's kind of a facilitated diffusion as well, right? So water, remember our other rule that we said right here is water follows salt. Right? So if we look at it, so if we have our salt concentrations, right? If we have 0.9% outside the cell, right, and 2% inside the cell, which way will water flow? Right? So water follows salt. So the solute concentration is higher in, which means water flows in. Right? So conversely, if we have pure H2O, right, and the normal concentration for salt in a cell is 0.9, the water flows in, right? And how long will it flow in, right? So it'll flow into the burst, right? And that's called plasmolysis. And this term lysis means to cut or break. So plasmolysis means it'll burst open, right? If we had a situation where we had our cell, right? We have our, oops, I can't get my pen to work. So if we have our cell, and our cell is 0.9% still, right? And I had a 1% solution out here, water would flow out. And what would happen to our cell? Our cell would shrivel, right? It would lose turgor, so it would shrivel. Okay, so osmoregulation, right? So if we have this here, is through salt concentration. Okay, so we talked about that, hypertonic, osmotic, hydrostatic pressure, and all that kind of stuff. And the last important thing we're going to talk about is active transport. So active transport is against, so the important thing here is it's against the concentration gradient. So if you're looking at an FRQ, how do you tell if it's active transport? So if we have a cell, right? If we have high concentration in the cell, we have low concentration here, and the substance still flows in, right? It can't be flown in by diffusion. It has to be active transport, right? So things about active transport, right? Requires ATP, right? So remember, ATP is what they call the currency it's the currency of energy for the cell, right? So it requires a carrier protein, but also requires ATP because I'm going from a low to a high concentration. An important reason, cell membrane potential. Okay, cell membrane potential is an important reason for ATP. So again, here, if you look at this picture here, right? If we look at that picture over here, we'll see that we have ions, right? We have ions here, and those ions are flowing in to an area of low concentration to an area of high concentration, right? And that only works because we have ATP working on it, right? Requires a carrier protein. So here we go, right? We have a carrier protein here that brings it to the ion channel, and then it moves it in, right? And ATP over here, right, powers the opening of the channel. Co-transport, right? Co-transport is simply using, it's like facilitated transport. It's using a second molecule for diffusion. And then we have bulk transport, right? So important thing for bulk transport is this word cytosis, or the suffix cytosis. I don't know why it does that. I'll get out of it. So let's try that again. So the important thing for this is this word cytosis, or the suffix cytosis. So cytosis means of the cell. Exo means out, so exocytosis is moving things out. Then endocytosis, endo means in, right? And remember, this is through vesicles, right? So it's moving through vesicles. And then pinocytosis is movement of water. All right? So super quick. And I'll talk to you later. Good luck on your FRQs, by the way.